Scientists at East Malling Research in Kent say they are now able to extract double the strength of a special product produced by the sweet wormwood or Artemisia plant to treat the mosquito-borne virus. The malaria-combating qualities of artemisinin, which is produced by the plant, have long been known.
Certainly the concept about the potential of a plant has been known since 340 AD. The Chinese were very much aware of this. They used it in their pharmacopoeia. It was listed. It wasn't overly clear the mechanism or what the active ingredient was, but it's been around for a long while. It was only very recently in the 70s that they became aware of the impact that it was having on malaria in places like Asia and China. And that's when the West became actually involved in trying to identify the actual chemical, the artemisinin, which was having this impact on malaria.
Artemisinin is found mainly in the hairy fern-like leaves, stem and clusters of the flowers in the new growth of the sweet wormwood plant, which can grow as high as 6 feet. The research undertaken at East Malling and its various partners has used £900,000 of funding from the Farming Ministry DEFRA to produce new plants which have higher artemisinin levels and can be grown in colder climates.
Our part of that is at East Malling is to optimise the agronomy and, as I've said, the irrigation that would deliver that. We have another partner, Frontier Agriculture, who are actually developing field growing systems so that you can actually plant this crop using seeds that are produced and a secure supply of seeds and actually grow it in Kent. And they, last year for instance, had I think 3 locations where they actually grew it as a field crop with different levels of success, but they've actually established that you can grow this in the UK with our breeding material and that of NIABs as a real agricultural crop. So we're a lot closer than we were 4 or 5 years ago.
Ethiopia switched to using artemisinin products as its first line of defence against malaria 5 years ago. The drug has proved to be 100% effective against the disease, compared with 74% recorded by quinine.
Well, the actual chemical, and it's called artemisinin, is a very complicated chemical, but we're getting to understand actually how it works and how it kills the malarial parasite when it gets into your blood. But it's produced in very specific parts of the leaf. If you look under the leaf of an artemisinin, there are very, very small glands. They're microscopic, and they're called trichomes. But within those trichomes, there's biochemistry going on, which is producing this chemical. So it's only produced in these cells. They're sort of like protuberances that hang off the bottom of the leaf. So they're very delicate. So you have to know and understand how to harvest the plant to make sure that those cells don't actually drop off, and you actually maintain the concentration of the chemical, because it's very volatile. So if you dry it too quickly, you dry it incorrectly, you'll lose the chemical. Clearly we don't want to lose the precious amount of artemisinin within those trichomes.
Researchers have successfully grown plants on 494 acres in southern England. New techniques to extract and purify artemisinin from plant material are being developed.
We're only interested in the leaves of the Artemisia annua, and it's in the leaves that you've got the tiny glands called trichomes, which contain the artemisinin. We dry the leaves from the plant and strip off all the stalks, because the stalks don't contain any of these glands, and we end up with just the leaves, which we mill, and we then put into an extraction column like this. And this is 19 litres, so we get about 8 kilos of milled leaves in this extraction column. And through the column we circulate a compressed gas, which is actually acting as the solvent to actually extract the contents of the glands. And the compressed gas with the artemisinin goes into a evaporator here, where the gas is recovered and reliquefied for recirculation, and then we decompress this to actually get out the extract of those glands, which is this sticky mess here. The difference between this process and other processes is the purity of this is much, much higher, making further processing a lot simpler and getting the active out in a much higher yield.
The aim now is to produce stronger stock plants better suited to the varying UK climate.
It's important that we have a system that's as efficient as possible, particularly if we're going to grow it in the UK, and encourage growers in the UK to invest in the plant material and to grow it as an economic crop, that we can have as high a concentration as possible. And I think there's great potential to carry on doing conventional breeding to actually further increase the concentrations.
It's estimated there are more than 1 million deaths from malaria annually in 109 countries. Scientists hope the solution to this major international problem can now be found in a weed.